Few things to get out of the way. I apologize in advance for speaking quickly. If I don't, we might only just solve the halting problem, and that would be a shame. Next, there's a number of affiliations up here. One of them is actually important. I still actually work for the White House. I've been working there for, almost 3 years now. So it's been a journey, I have to say, as of late. And this comes with a disclaimer. So I'm required to put this before every talk that I give, which is that this is a personal talk. This talk may not reflect the views of the president or the administration. But what I can tell you is that they used to. And if you want to know official White House policy these days, you can find it on this cool site called Twitter. All right, so with that out of the way, let's talk about the war on air. So I think we can all agree that error in code is bad, right? We don't like error. So there's code for software, which I think most of us are used to dealing with. And when we get errors in code for software, really awful things happen. So if you get it wrong in the rocket explodes. That's bad. And we get it wrong in lots of other stuff. You get vulnerabilities like Heartbleed, which can lead to billions of dollars in damage as a whole. So errors in code for software, that's bad. Errors in code for people is also bad. In this case, I'm talking about genetic code. It's just another kind of code. By the end of the talk, I hope you'll understand that it has a syntax and semantics just like the code that you and I typically write when we're programming. So when you get errors in code for people, you get rare genetic disorders. You get predispositions for a handful of diseases. And you sometimes get, in fact, even oftentimes get stuff like cancer. These are all errors in genetic code that lead to these things. And I'm going to argue that we can fix both kinds of errors in code. And to the extent that I can draw a unifying theme at all between these two kinds of error fixing, I would say that it's approximation, that we can use notions of approximation to find ways to fix errors in code for people and errors in code for software. Now, they're very different kinds of approximation, but it's still approximation. So the main ideas for this talk are as follows. Let's talk about software briefly first. Let's suppose that we could solve the halting problem. So if we could solve the halting problem, if we had some magic function that told you whether or not some program for some input halted or looped forever, I assert that you could wipe out the vast majority of software errors that are out there today. And I'll go into some detail about this in just a moment. But of course, we can't solve the halting problem. So what we're going to do instead is approximate solutions to the halting problem. We're going to use a technique called abstract interpretation. I'll walk you through a very specific technique called abstracting abstract machines that allows you to, again, approximate the solution to the halting problem, or really approximate the behavior of a program. And the short version of this, if we really were just approximating the halting problem, the one way to see it is instead of saying, yes, it halts, no, it doesn't, you have a third option. You can say, I don't know. Because you can always say, I don't know. And now the halting problem, or the approximate halting problem, is trivial to solve. You can just return, I don't know all the time. All right. So let's talk about people. Well, so for people, I argue, if we look at their genetic code and we isolate the errors as the cause of their disease, then we can also find an optimal solution for them, some ideal pill or treatment that they can take that will serve to correct for or account for that genetic defect. Someday we'll be able to go in and do like live patching, and that someday is not even that far away. But for right now, using even today's technology, we can use pharmaceuticals to account for errors in genetic code. So what we want is a function that goes from people, or really their data, into a pill or a procedure that works for them. And of course, this function is also incomputable, but we can approximate this as well. And so instead of giving you back a single pill you should take, it'll be a set of pills that might work for you, or potentially some experiments you need to run that will get you closer to finding out the pills you need to take. So let's talk about fixing software first. So let's just also agree up front that software is terrible. And what I mean by that is like, when we look at like the state of software today, we not only have vulnerabilities, we have vulnerabilities with their own brands. And brands and product lines even. I mean, this is the state of software in 2017. And it costs billions of dollars, and we just sort of go, whatever, that's fine. And pretty much everything is hackable now. And in fact, I was even surprised to learn that even restrooms these days need information security. But if this surprises you, all that really means is that you've just never been to Japan. Because I guarantee you can hack these toilets. So why is it that software is just so awful? Why is it so terrible? Well, there's been some detailed analysis out there about why software is awful. So yeah, C programmers. So what are some solutions to the problem, to that specific problem? The fact that we keep writing code in legacy languages like C and writing all these terrible vulnerabilities. Well, what we need to do is we need to engineer software. And it's interesting because there's this field of study called software engineering. But I would argue that software engineering is not really engineering at all. And I apologize to probably half of you in this room that are software engineers. So with that in mind, why do I say that? Well, an engineer uses prediction. If you're a real engineer anyway, use predictive models. And so for example, if you're doing anything classical engineering, you have like F equals ma. And so from force, you can go to acceleration. Through acceleration, you can go to velocity. From velocity, you can go to distance. And now you can sort of predict exactly how a particle's path will evolve. And if you are using this as an engineer, and say designing the targeting system on a battleship, you can use this to very precisely predict the path of a projectile if you're going after some enemy battleship. And that's actually quite useful. However, if you're a software engineer, things don't always work out quite so cleanly. And a good example of, I think a better analogy even for software is if we look at how we build bridges versus how we actually engineer software today. So, and I'm always reminded, or I was instantly reminded when I was taking software engineering as an undergrad of this Calvin and Hobbes cartoon right here. Because they're driving across a bridge, and Calvin asks his dad, "Hey, Dad, how do they know what the load limit on a bridge is?" And his dad very smartly replies, his dad is clearly a programmer, says, "Well, they just drive bigger and bigger trucks across the bridge until it breaks. Then they weigh the last truck and they rebuild the bridge, because that's how you know." Well, the thing is, okay, that's obviously not how we do it with bridges, but this is exactly how we do it with software. We just drive tons of test cases until we break our code. But if you're an actual engineer building a bridge, of course you have static and dynamics, you have Newtonian tensile strength models, you have all kinds of stuff to predict how a bridge will behave under a continuum of circumstances, that you know exactly what a bridge will do every time something drives across it. So, can you predict software? Can we take software engineering and make it a real engineering? Can we apply the same principles of prediction to our field? Does anybody know whether we can do this or not? No, we can't do this. And it was Alan Turing who ruined it. So he gave us the only commandment of computer science, that you cannot decide the halting behavior of a program. And because of this, all the fun stuff we'd like to do about predicting program behavior also becomes unsolvable. What a bummer. So the way I like to think of it is that, whereas physicists have black holes and event horizons beyond which you cannot see, in computer science we have loopholes. And it's really loops and recursion that drive the difficulty in reasoning about program behavior. These are the things beyond which we cannot reason automatically. So what can we do about this? How do we solve this problem? Well, there's several things we could do. As I mentioned earlier, we could use better languages. We could stop using C and C++. That would be great. We could move on from this 1960s, 70s technology onto something at least 90s era. Or perhaps some vastly superior alternate universe altogether, where we just can't even write errors in the first place. That would be interesting. Okay, and actually, part of my life as a computer scientist, which ended 5 months ago, I did some research for DARPA, where we actually sort of took this as a theme and said, can we come up with better ways of doing programming so that we don't write errors in the first place? That was the theme of Crash. So I could give a talk about that, but I'm not going to give that talk today. Or what we could do instead is we could solve the halting problem. So I think this is actually easier. So we'll just solve this, and what we're really going to do is approximate solutions to the halting problem and use these as if we could actually solve it. And to do this, what we're going to do is we're going to play the static analysis game. So what do I mean by the static analysis game? Well, I want you to imagine or visualize the execution of a program as a sequence through some state space over time. It's just state, 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 all the way through its execution. So what we're doing when we play the static analysis game is we obviously cannot predict this trajectory exactly. That's solving the halting problem. But what we can do is put upper and lower bounds on this path. So we can define sort of the region of the state space through which it might traverse as it executes. So then what we can do is we can look at bad regions of the state space, places where we're overflowing integers, places where we have buffer overflows to reference signal pointers, places where we have malware, and we can say, okay, well, those are the regions we don't want our program to ever go. If you look at the bounds we computed here, it's not quite tight enough to rule out malware, and it's not quite tight enough to rule out buffer overflows. But if we come up with a tighter approximation of the trajectory, it actually is. And now we can say that, look, no matter how this program evolves, it doesn't go to the bad places, so we trust it. This is the static analysis game, coming up with ever better approximations of your program's behavior without actually becoming incomputable, or, of course, intractable. If you go deeply exponential, that's no good either. So how do you play this game? Well, there's a general technique called abstract interpretation developed by the Couzeaus back in the late 70s. And I'm going to show you a particular way of doing abstract interpretation called abstracting abstract machines. This is a way of building a model of a program's behavior so you can essentially model that trajectory or bound that trajectory in some way. So the original publication on this is from me and David Van Horn 7 years ago now. Oh, my God. And it's an ICFP. But what I really recommend if you want to know more about this is read the journal version, which I so lovingly put so much effort into, but nobody does read, because what I did here is I put all the math side by side with transliterated Haskell code. So it's an executable paper. You can just take stuff out of this paper and run it directly in Haskell. And so now you can actually follow along directly with the executable model of the paper. So what's the core idea behind abstracting abstract machines? Well, you take your interpreter for your language, and you pass it through this process called abstracting abstract machines. And at the other end is not an interpreter, but an approximator. And now this approximator can predict for you how a program will evolve. This scheme requires a very specific structure to the interpreter before you do it. And the structure is as follows. You take your program, and you have to have some sort of injection function that gets you into the concrete state space for this interpreter. So you take your program, inject it into a state space. Then what you do is you have a step function that takes a state, gives you another state. So now you have a model of execution for your programs. Here's an example of a particular step function. This happens to be for an abstracted version of Dalvik. I'm not going to go into the details of any particular semantics today. I just want to show you that these things exist. So then what you do is you take your program, you inject it into your initial concrete state, and then you just step, 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 step. That's your model of execution for the input to abstracting abstract machines. Now, if you could actually do this, if you could just trace out all these states to which a program might ever go, you could then ask, is there a vulnerability in any of these states? And if your program terminates, great, you can answer this question very easily. But if it doesn't, like many programs might, then there's no way in advance of knowing exactly what your program's going to do. You can't just map out an infinite number of states. You can't just do that. So what we have to do is we have to abstract in some way. So to abstract and make this a tractable problem, we're going to approximate, and we're going to do a finite abstract execution of this program in an approximating state space. Now, some precision will be lost. That's the price you pay for getting around the halting problem. You're going to lose some precision. To do this, we come up with an abstraction map that takes us from the concrete state space into this finite abstract approximating state space. And this map is going to be maintained under a new way of executing the program. So if we start off in a correspondence with some concrete state and an abstract state, and we take a step in the concrete interpreter and this approximating abstract interpreter, then this abstraction relationship is going to be maintained in some way. So that we know that no matter what path we take through the concrete execution, we're always going to maintain this correspondence and have a representation in the abstract world up there. Now, if you're being very nitpicky about this, it's not really a map. It's actually a composition of a map with some sort of partial order, because we can actually have states that approximate other states, and we're allowed to arbitrarily widen the precision of a state at any given point. But that's not necessarily a particularly important detail. So how do we do abstracting abstract machines? Well, it's a multi-step process, and the first step is to diagnose the problem. It's to figure out why if you just take your program and run it, it might not stop. And we've sort of already talked about this. If you just have this machine that's running, there's a reason it may never terminate. Any of these individual states could be unbounded. There's an unlimited number of states through which you could traverse. And if you look inside the state, or typical state for most languages, you'll find the following 4 components. You're going to have the instruction you're on, you're going to have the frame pointer, or some model of the environment, you're going to have the heap or the store, or and the stack or the current continuation. And 3 of these components can be unbounded. You could have an infinitely large heap, infinitely large stack, or an unbounded number of frame pointers. So that's the sort of the core problem. If we want to get down to a finite state space, these are the 3 areas we have to attack, because that's the source of the unboundedness. That's what we have to break in order to build an approximator. So the key step is step 2, which is to abstract, or really to finitize in some way. We're going to take that concrete state space and make it finite. So how do we do that? Well, I'm going to use a model of computation called a CESK machine. This is actually due to Fleisig and Friedman. Actually, Dan Friedman's sitting right back there. So this is a model of computation that they developed almost 3 decades ago now. And it's still valid, actually, for modeling the vast majority of programming languages we want to model today. So you can take almost any programming language, and you can model its semantics using a CESK machine. And there's 4 components here. You have a control component, an environment component, a store component, and a continuation component. So I'll step us through a CESK machine now in a little more detail. And then we're going to figure out how to make each of these components that are unbounded, bounded. So the control component, if we're dealing with something like a functional language, it's going to be an expression. It could also be a statement if it's not a functional language. The environment is a map from variables to addresses. So this is sort of a two-phase lookup machine. You have to go from variable to address, and then you go into the store to go from address to value. And then we have the stack, or the continuation. And the stack is an unbounded sequence of stack frames. So there's multiple sources of unboundedness here. Addresses are unbounded. The number of stacks is unbounded because we could just keep pushing frames forever. And of course, values themselves are also unbounded because values include things like integers. So there's an unbounded number of integers. So we have to come up with some way of approximating these three components. We'll start with values. So let's assume in a very simple language that all we have are integers and objects. Well, integers are pretty easy to abstract. We can finitize these in a relatively straightforward fashion. We'll take all the integers and we'll just smush them down. So instead of having out to negative infinity and positive infinity, we'll just have negative, 0, and positive. And then any integer can be represented, any abstract integer can be represented as a combination of essentially their polarities. And once we have this, we can model arithmetic on this abstract version of integers as well. So I won't go into detail into that. Just assume that you could figure out the details on your own. So now what we're left with is approximating objects. And an object is a pairing of a class and an environment. Or if we're dealing with a functional language, then what we're really playing with here are closures. And a closure is a lambda paired with an environment. So the exact kind of approximating process applies here as well. And so we have a finite number of classes for any given program. And then what we need to do is come up with an approximation for environments. Because what we're doing for field lookup is we're going from the field name to an address for that value. So once we're going to assume, however, that we're going to approximate environments some other way. So if we assume that we have a bounded number of environments, then of course we've bounded objects as well. So now we have an abstraction for values. So we need to go back to our whole state space and pick out the next thing to finitize. And in this case, that's going to be addresses. Because if you finitize addresses, then you've also finitized environments. Because the only thing that environments depend on are addresses. So how do we finitize addresses? Well, we just declare them to be finite. That's it. So we go from an unbounded set now down to some arbitrary finite set. And we declare that there is an abstraction from concrete addresses to abstract addresses. And actually, this is probably one of the most interesting parts of the talk, because playing around with how you do this abstraction, how you partition up the division between concrete and abstract addresses, this determines the complexity, the speed, the precision. Everything about the analysis is driven by this choice right here. 2 decades of research in static analysis can be summarized as playing with this function. I'm going to skip over that entirely. So we now have a finite set of addresses. In the extreme case, you can use a singleton set. You can have a single abstract address. It's not going to be a very precise analysis, but you'll get an analysis. So with that in mind, as soon as you have a finite set of addresses, values are going to start merging because they're going to start sharing addresses. You're going to come to a point where you're going to do a malloc, and we're not using C really. And it's going to say, oh, that address is already in use, but you're going to assign it anyway. And now 2 objects have to live at the same location. So that's why the range of the store now expands to allow multiple values to live at any given address. In this, now you can see where some of the imprecision is coming from. If you look up that address now and say, what object are you? Says, well, maybe I'm this one, maybe I'm this one. I don't know anymore. And so that's why things start to get imprecise when we finitize. But all this really leaves at this point is continuations to finitize. So what we'll do is we'll say, well, we'll do a little refactoring. We'll say, well, that this unbounded list of, or sequence of stack frames, you can think of it as a linked list of stack frames. So and when we look at it this way, we can say, well, all we have to do now is cut this little recursive knot between continuations and continuations. To do that, there's a technique in abstract machines called threading something through the store. So we're going to store allocate continuations. There are actually languages out there, or implementations of languages that do this. So like SMLNJ uses heap allocated continuations. It actually does that. And we're going to essentially do the semantic equivalent thereof. And the reason we do this is that it pushes the unboundedness of this stack into the unboundedness of addresses. And so now, as long as we can finitize addresses, we can also finitize the stack in some way. So to go back to the semantics, the little surgery we have to do here is we put continuations in the store. And now addresses and vowels and continuations live side by side. When you want to look up the next stack frame beneath something, then you go ahead and look up its address. And of course, now, when you want to return to something, when you're actually looking at its continuation, you might return to multiple continuations. So you can get confusion on return flows. This is something that's just going to happen when you do static analysis. All right. I'll show you a fix for that in a second. All right. So now we've actually finitized everything. So now we have this finite abstract state space through which we can execute this approximator and figure out what these programs are actually doing. So we have an abstract machine. And that's the essence of abstracting abstract machines. Just go through this process and then play it over for the semantics. So the semantics go from this through the machine into something that looks like this. And again, the specifics don't matter for today. The point is that there's a process you follow, a mental discipline that you go through in the state space and in the semantics for turning this into an approximator. So if you want to know a whole lot more about this, you can watch a recording of a talk I gave that corresponds to a blog post I made, which goes through how to build an abstract interpreter following this exact process for a very simple register transfer language in about 80 minutes. So I just live code the whole thing right in front of you that steps through everything I just talked about. So the question now is, let's assume that I've just showed you how to solve the halting problem or play the static analysis game. The question is, can we win it? Can we actually do something meaningful with it? And I would argue that the answer is yes, and I have some proof that this is true through another DARPA program I participated in called APAC, or Automated Program Analysis for Cybersecurity. And this was really predicated on a terrible idea. Not my terrible idea, somebody else's terrible idea. So the idea was, let's go ahead and give every soldier in the US military an Android device. And let's let them download like the Drone Strike app, so they can call in drone strikes or whatever they want using their Android phone. This is obviously a terrible idea because I have like this open app store with open apps and anybody can submit whatever they want. So they at least recognized that that was a bad idea. They said, okay, well, this could be bad. You know, we could have people who are leaking their locations to enemies through these malicious applications and stuff like this could happen. So let's not allow that. Let's have an app auditor that magically figures out what's safe and what's not. And so good apps come in, they pass through the auditor. If it's valid, after some sort of scan, then you say, okay, you go in the app store. You're safe. You have no malware. Then, if some malicious app from some bad actor goes in, we say, no, you're bad. You're malicious. You cannot go in the app store after you've been scanned. You go out. So that's the idea. That's what this program is supposed to build, was a way of building this auditor for good and bad apps. So we signed up to do that. We're like, yeah, let's go ahead and do that. Why not? Let's see if we can figure out what's good and what's bad. And to do this program, every 6 months, we had to compete in an engagement with red teams where they would throw apps at us. And we would play App Store, and we have to figure out what's good, what's not. If we said it's bad, we also had to say, and here's where the malware is. This is what's been hidden in this code. So 6 months in, at our very first engagement, our accuracy in determining what was friendly and what was not was 0%. It was like total failure. And I remember going to that PI meeting afterwards and looking at the program manager who'd just given me all this money to do this. And I go, you know, sometimes there's a difference between the vision that you put in a research proposal and the deliverable that you end up with at the end of the day. The animated GIFs placated him for at least 6 months, you know. But he's like, no, next time it actually has to work. So by the end of this, over a number of years, we actually got better and better and better at doing this. We did a ton of research in static analysis to make this incredible tool for actually finding malware in code. And for the last 2 engagements, our accuracy was actually 100%. You know, we got everything exactly right. This to me was sort of some empirical evidence that, hey, maybe we can do this in practice. Maybe we can play the game so well that, you know, for all intents and purposes, we can solve the halting problem. And all this research that we did, some of the key innovations, if you really want to know what it was, one thing we did is we broke away from using finite state models of approximations to pushdown systems. So pushdown systems are still not yet Turing complete, and so even though they have this infinite configuration space, you can still compute lots of interesting things about them statically. So the key intuition for how we did this is we took the continuations out of the individual states and we put stack changes on the edges to say that you're pushing a frame, popping a frame, or doing no stack change at all when you transition from this state to this state. And so we built up finite graphs that look like this, essentially. We have published a number of papers that established this and then used it going forward. We also figured out how to make these approximators go really fast. So we published a number of papers on optimizing abstracted abstract machines. And today, we can actually get these things to be reasonably fast as a result. We figured out how to analyze parallel programs. We also figured out how to analyze programs in parallel, including even on GPUs. And we used some other tricks, too, like using SAT solvers to speed up the computation of these analyses as well. So we did a lot of tricks like that to make them go pretty fast. We also started layering more interesting analyses on top of it. So there wasn't just building a model program behavior, it was also telling you something about the program too, like computing information flow analysis that's saying, it's not just this kind of information, or it's not just this information, it's this kind of information, like this came from the network, or this came from this sensitive data source, and this is where it flowed to. And then we combined all this together to do actual principled malware detection by the end. So We now have a follow-on project for this, another DARPA program called STACK. We're going after even richer kinds of bad things, richer kinds of vulnerabilities. So now we're not looking at just, we're looking at sort of regions of code that could themselves be malicious, not just individual points in the code. And by that, we're really going after things like side channel attacks and complexity attacks, where malicious inputs could lead to this exponential explosion in behavior. So now we'll analyze regions of code, we'll summarize them with things like recurrence relations, and then use recurrence relations solvers to say this region of code has complexity like this, and that could be exponential, therefore there could be one of these vulnerabilities right here. So that's what we're doing in this follow-on program. And we're, again, it's basically the same pattern, got 0% on the first engagement, and we're moving up from there. So, I think there is hope that even things as challenging or as difficult to reason about as complexity attacks could fall under the auspices of auditing under static analyzers. Same for space attacks and even side channels. So, in conclusion, I think, at least for the halting problem, Alan Turing might wish to revise his statement if he were around today. He might actually say, well, maybe you can solve the halting problem sometimes in practice when it actually counts. So, with that in mind, let's move on to cancer. So what about people? How do we fix errors in people? And I'm going to argue that we can. And a lot of it is actually thanks to this guy, who really kickstarted this whole movement for precision medicine in the US. So he launched this precision medicine initiative 3 years ago. And precision medicine, to me, is all about fixing errors in people at the level of genetic code. So what is precision medicine, actually? And how do we fix these errors? Well, I would say it's really data-driven medicine. So if you're using data to optimize the healthcare of an individual patient, you are doing precision medicine. And the piece of data that everyone's super excited about right now is the genome. We're now considering how do we do healthcare in the context of knowing your genome. If we know the blueprint for you or your cancer, what more can we do that we couldn't do when we didn't know that? And at the end of the day, what we're trying to do is deliver the right drug to the right patient at the right time. And that was the mantra that Obama kept repeating over and over, working for him. That's the essence of precision medicine, right drug to the right patient, right time. So how do we cure cancer? Well, I'm going to argue up front that cancer is not a disease. Cancer is, in fact, many diseases. And in particular, it is many rare diseases. So using reduction like a good computer scientist, I'm going to say that we don't really need to cure cancer. What we really have to do is cure rare diseases. And if we can cure rare diseases, then we can cure cancer as well. So first, recognizing that this is not a room full of biologists, let's do a little biology 101. So to begin, DNA is a char star. And in this language, we have 4 letters. We have A, T, C, and G. These correspond to the 4 nucleotides that make up these cute spiraling double helixes that is your DNA. So you've got your DNA, and you've got regions of your DNA that correspond to genes, and these undergo a process called transcription, where your DNA is transcribed into another language called RNA, which is A, U, C's, and G's. And then RNA is paired off after splicing, which we're not going to go into, in these little 3 nucleotide sequences called codons. And each of these 3 nucleotide sequences, or codons, is then translated into an amino acid. So there's a translation process here. You do this for the entire RNA sequence, and then these amino acids, which are linked together, fold into 3-dimensional space into a protein. At this point, there's usually a number of modifications that happen after the translation of the protein, which is why we call them post-translational modifications. Sometimes you'll attach a sugar, sometimes you'll phosphorylate proteins. There's a whole bunch of things you can do to a protein once it's been produced. But that right there, that's like basically all of biology now. So now you're biologists. The other thing to realize is that many proteins are enzymes. And enzymes actually have a semantics to them. So an enzyme is something that turns A's into B's. Sometimes it takes 2 things and makes them one thing, or many things and makes them into one thing. Sometimes it goes the other way. Sometimes you take one thing and you make it into many things. And so the genome has a syntax, and it actually has a semantics too. So DNA itself is kind of an instruction set. We can even examine what that instruction set looks like very precisely. So for example, if we take this sequence, ATG, GCC, TGA, we can reinterpret this in terms of a program. First, we break it apart into constituent codons, and then we look at what each of these codons is saying, telling you to do. So ATG is saying, ATG is how you start. It's begin construction of a protein and insert a methionine. So methionine is always the start instruction for building a protein. And then, if you look at GCC, this is a relatively complex codon that means compile a C program. Or in reality, it means insert an alanine. So this is insert alanine. And then TGA is a codon that means stop. or end construction of this protein. So taken all together, this sequence really means insert a methionine, insert an alanine, and stop. And then eject that protein, let it fold, and do its thing. So now we can understand what mutations do in the context of this program. So for example, if we take this program and we change it into this program because we have a mutation, well, now it's a different program. So instead of inserting a methionine and then an alanine, that alanine is now an aspartic acid. And maybe that changed what the protein did, and maybe it didn't. So because a lot of mutations are benign. They don't actually change the behavior of a protein. There are large regions of most proteins where they're somewhat insensitive to the particular amino acid there. But there are some regions of the protein where they're very sensitive to that specific amino acid. So some will destroy function. Some will actually increase function. Sometimes increasing function is bad too. And some will change function entirely to something else. They'll make it do a completely new thing that it couldn't do before at all. But most of the time, I would say, when you get a mutation, you wipe out the function in some way. That's probably the common case. So rare diseases actually, in many cases, are just a single mutation driving the disease. One mutation somewhere in the genome creates an entire disease. So I think the best way to explain this is through my favorite model organism, which is the stick figure. Some people like fruit flies. I like stick figures. So the reason I like stick figures is they only have 6 genes. So if you look at a stick figure and you look at the effect of a mutation that decreases the activity of gene C, well, it's going to do something to it. In this case, you've just learned that gene C is linked to head size. So what we're doing for this rare disease in stick figures is we're looking for something that reverses the effect of the loss of function in C. And hopefully if we find that, if we find a drug that does exactly that, then when we go back to our model organism, we see an increase in head size at that point. So that's the game we play in even rare diseases for humans. We find the defect in terms of the gene, and we say, okay, well, let's undo that effect. If it was a gain of function, let's find an inhibitor. If it's a loss of function, let's find an activator. And that's sort of what we do. So the key thing you have to recognize now is that cancer is multigenic. So with cancer, you're never fighting just one mutation. There's one mutation usually initially. Maybe that was a tumor suppressor that then led to a cascade of other mutations. But cancer is a pretty complex beast. And so you might have to target multiple mutations at the same time and find compensators for 3 different genes all at once, say, or 2 or 5. And then as soon as you found compensators for all of these, yet another mutation pops up, and now you have to fight that too. So fighting cancer is like fighting multiple rare diseases all at once. That's because tumors are evolving. When you're fighting cancer, you are fighting evolution itself. That tumor is fighting against anything you do to it to stay alive. And so this, I think, allows me to explain why cancer can be so difficult to treat. So if you start off with one population of cancer cells, and they evolve over time, you actually end up with lots of different subcolonies there, which have entirely different cancer genomes because they all continue to mutate. Now, if you apply a drug and to wipe out this particular cancer, it might be effective against 99.9% of all those cells. But just by luck, there might be some of those cells, because of how they mutated, that had a natural resistance to whatever medication you applied. So they survive. And now those things proliferate. And when they proliferate, they pass on to their descendants the resistance to the first medication you tried. So now if you try that same medication again, it doesn't work. They all know how to beat that by now, because that tiny little population that survived, it knew how to beat it just by luck. And now it survives. That's why you're fighting evolution itself when you're fighting cancer. So curing cancer really is like fighting multiple monogenic diseases on the fly. Because again, it's always evolving against you. You always have to adapt it and fight against it. So how do we cure monogenic disease? Well, I assert to do this, we need an algorithm that maps us from mutations ultimately into pills. So I will sketch out this algorithm for you very quickly as a flow chart right here. I'm not going to go into a whole lot of detail on it. I will then sort of dive into some cases and show you exactly what this is doing, or how we do it. So what this algorithm is really branching on is it's saying, okay, well, let's isolate the mutation. Let's find what's really driving disease. And let's figure out, is it gain of function, loss of function, total loss of function, change of function? What happened here? And it's providing either things you can do in terms of medications you can take, or it's providing experiments you can run that will highlight the next set of things you need to do in order to figure out what the pill you need to do is. So the question is, will this algorithm that I've just presented actually work? Can you actually use this to find a pill for any given mutation? The answer is yes. The question is then, how do I know that? I'm a computer scientist. I have a grade school education in biology. Why am I telling you any of this? Well, now I have to tell you a personal story. So this is my son Bertrand. He's almost 10 years old today. And when he was born, we had no idea what was wrong with him. So he was having seizures. He had a severe developmental delay and a movement disorder, and for some reason, a very curious lack of tears. I mean, he could cry, but emotionally, he could feel it, but no tears would ever come out of his eyes. So he had this very strange constellation of symptoms. And this was our life, for 48 months. It was trying to figure out what is going on with Bertrand. And in the course of this, I realized very quickly that I needed to learn genetics because this is a genetic disease. So I learned a lot of genetics, and I realized very quickly after that we needed to use genome sequencing to figure out what the root cause of this disease was. And so that's what actually ultimately happened. We did trio sequencing on both parents and Bertrand. And from this, we found out that he had inherited 2 loss-of-function mutations in a gene called NGLY1. And we believed that this was the cause of the disease. But along with this diagnosis came some very interesting news. Because this, by the way, this was like 6 or 7 years ago. So this was very cutting edge at the time. You couldn't do this outside of a research project. Now you can do this clinically. But back then, this was research. The researchers we were working with said, this is actually the first case we've ever seen of this disease, if this is what it really is. It's the only case anywhere in the medical literature, so in many ways it's an N of 1. And so the question is, well, how do you know that, because we all contain tons of mutations. How do you know that this is really the mutation driving this disease? Well, to do this, you have to do a really deep analysis of the gene. And this actually can get quite involved. Now, we were lucky that this is a research project, and there was mechanisms for actually diving into the cells here and figuring out if this is really what was going on. So to understand, the thought process for this, you kind of have to understand what NGLI1 or NGLI1 is doing. And NGLI1 is a gene that encodes an enzyme called N-glycanase. And N-glycanase is responsible for deglycosylating misfolded glycoproteins after they've been retrotranslocated from the endoplasmic reticulum into the cytosol. So obviously, what's happening here is we're missing a recycling enzyme. And if you take away a recycling enzyme, you'd expect to see an accumulation of trash. And there was some evidence that maybe there was trash inside Bertrand's cells that maybe could be an explanation for the cause of this disease. But of course, this process, you can't do this every single time you see a tumor, every single time you see a patient. You can't go do deep fundamental science. You have to have a different way of interpreting the meanings of mutations, because it just doesn't scale. And so we need to work around, and that's where this guy comes in. President Obama announced the Precision Medicine Initiative almost 2 years ago, actually 3 years ago now. And what it's doing is it's laying the scientific foundation, or really building the Rosetta Stone for the human genome. And the way this works is it's recruiting 1 million Americans and asking them to donate their electronic health records and their genomes to science, and to build a massive scientific database so that we can start to infer the relationships between genes, disease, and environment. So in some sense, we're building a genetic telescope. And what this telescope can then do is it can peer into any region of the genome and say, if you have a mutation here, what is this doing to the health in this cohort of 1 million Americans? For the people that have a mutation here, what is their health like compared to everybody else? So this database really will unlock the meaning of all these mutations floating around in your genome and tell you, how it contributes to disease or predispositions to disease. When I finally got to the White House, I actually didn't work so much on that. I worked a lot on that before I got to the White House. And when I got to the White House, I realized that the fastest route to building that Rosetta Stone was actually not through this new program, but through an older program called the Million Veterans Program, which had already enrolled over 500,000 veterans to do essentially the same thing. And veterans are, for all intents and purposes, mostly like the rest of us. And so we can use their genomes as this guide. So the one thing they didn't have was proper computation. So I got... or help pair up the Veterans Affairs with the Department of Energy, so we could use Department of Energy supercomputers, or an unplugged cloud, as it were, in the end, to actually build a genomics research environment, which actually launches on November 17th, to allow anybody to sort of start running these queries over this data set and say, well, what are these mutations doing in terms of human disease? What are these genes doing in the context of human disease? But what about Bertrand's mutation? This database didn't exist back then, so how do we figure it out for him? So to do this, I wrote a blog post. This was the way I short-circuited the entire interpretation process, because my reasoning was, if another patient exists with this, and they have the same mutations in NGLY1 or similar mutations in NGLY1, that immediately means this is the root cause of this disease. So this is the blog post that I wrote, and it was really designed to do 2 things. So it had to go viral. That's why the title's there. That's why Liam Neeson's there. Could have been a cat picture too, probably would have just as well. And then there's 5,000 words down beneath this for search engine optimization, so that this thing would rank highly in Google search results and all that. So the crazy thing is it actually worked. So it did go viral, because y'all are pretty gullible. And sure enough, it started ranking very highly for all the right Google search terms, so that if a patient or a parent were out there searching for whatever their child had, they would find this page and they would find us. What I didn't expect is just how fast this could happen for a brand new disease. So within 2 weeks, a sibling pair in Turkey was already in touch with us through their physician saying, we have exactly the same disease. They had done sequencing. They had this list of mutations. They were going, gosh, I don't know which of these is the cause. So NGLI1 was on that list. And when that doctor saw this blog post, he goes, oh my God, that's the disease. That's what this is. And so that was the confirmation that NGLI1 was the true culprit for Bertrand. So, and what this really did was, because we started finding people right away. People started showing up all over the world, and it's been 5 years now. I think we have 60-some patients. What it did to have this community almost overnight was to really wash away that dark night of uncertainty you face when you only have one patient and replaced it with the sunlight of science and community. And that community today is very active and united in actually targeting the root cause of this disease in terms of understanding, but also finding treatments and therapies that'll work for it as well. If you want to know more about this whole process, there's an article in the New Yorker that describes this process of using social media to find more patients. It's a process that's actually been repeated several times over now and something that I help other patients do. The other question is, can you find a treatment? Now that you understand the root cause of a disease, can you attack the root cause? Can you find a pill that actually works? So through some investments in basic science, I actually came to a much better understanding of what was really happening in Bertrand's cells. We know that in the absence of this recycling enzyme, that trash starts to pile up. But this trash is piling up in a very specific way. And I predicted that it was actually sequestering one particular metabolite. That metabolite is represented here by this little blue square. So this little blue square happens to be called N-acetylglucosamine. And you can, if you Google it, you'll realize very quickly you can buy it on Amazon. And for what should be therapeutic quantities, or so I reasoned, it costs maybe 25 cents a day. It is not particularly expensive. So then it's now it's like a Star Trek level ethical dilemma. Do you give this to your child? Because you can buy it on the internet. So, so I bought it on the internet, and I took it myself just to make sure it was safe. And I didn't die. I'm here today to tell you that. So that for me was essentially FDA phase 1 safety testing. And this was where I have to remind you that although I'm in government, I am not the FDA. And don't do this at home. So, but it wasn't clear if it was ethical to give it to Bertrand. But that was actually answered very quickly by Bertrand himself. He went to the hospital with a very severe pneumonia, which he sometimes would get, and came very close to dying. And I remember sitting there at his bedside, sleeping there, thinking, if he had died in the hospital and I had never tried this thing sitting on my shelf and it could have made a difference for him, there's no way I can forgive myself as a parent. So once he pulls through, once he's healthy, I'm going to put him on this compound. We're going to see what happens. So that's what I did once he was out of the hospital. I put him on this compound. And about 3 days after he was on this compound, in a disorder where no child had ever cried a tear, he cried the first tear of his life. And the same thing actually happens for all the patients that go on this compound. They all start to cry tears. So it was, as a parent, I think I had the same reaction we all did when we saw our child cry for the first time, which is I collected the tear, I packed it on dry ice, and I shipped it to a lab in California for analysis. And that one small tear from Bertrand, of which he's cried many more since, really ended up playing the role of an ocean of science for the disorder as a whole. That unlocked a lot of the mechanism behind this disease. And we knew at that point, okay, we can hit this thing. Because actually it does better than just giving tears. It also takes away his very strange nighttime seizures. And so you don't know what's going to happen when you do something like this. You don't know what to look for. But these were very clear signatures that this was actually working. Now, you're a computer scientist, so you're probably satisfied at that point. But when I give a talk like this to biologists, they always go, well, that's great, Matt. I'm glad it works in people. But we're biologists. What we want to know is, does this work in flies? And well, I guess we could figure that out too. So we took some flies, and we gave them this disease. And we said, okay, well, what happens? Well, they die. You have about a 17% survival rate once you give them the fly version of this disease. And then we started feeding them this compound, and whoa, their survival rate jumps to like 80%, 80, 90%. So, okay, looks like it works in flies too. And turns out you can do a lot with flies that you can't do with people. So we understand a lot about what's happening from this, just from the flies. And in fact, if reviewer number 3 would just let some issues go, we will have a paper about this out probably in the next few months. That explains a lot of what's really going on with this particular compound. And in general, okay, so how do we do this in general? How do we do this sort of again and again, where we say, well, what's accumulating, what's in deficit for disease? How do we modify a patient's diet or metabolism to account for that? Well, this actually can get somewhat computational. So you can take these rules for metabolism and you can look at them as almost like rewrite rules. In fact, they essentially are rewrite rules, but on graphs. You can model these in a language like Kappa, which is kind of like a functional programming language, except its syntax is terrible. And You can then take out a rule that represents a missing enzyme for a genetic disorder. Then you can say, what happens to this new system? And so I've actually partnered up with Will Bird in the back and Jean Yang at CMU, and we are using this language to model human diseases and what happens when you take away a particular enzyme. So here's actually the very first disease we ever modeled. This is a disease called PKU, and we could show that when you take out this particular enzyme, you start getting an accumulation of something, which in this case happens to be phenylalanine, and you also get a deficit of something, which in this case happens to be tyrosine. And so now we know that for PKU, or we predict for PKU, that you eliminate phenylalanine from the diet, and you supplement with tyrosine to normalize. We could even simulate the effect of administering a treatment, too. So the question is, what happens now? I wanted to keep going, and I realized that I think we actually can. So I said, well, let's go from computer science deeper into biology and medicine. And this is where I started becoming less and less of a computer scientist and more of a biologist. So I actually got a grant funded to do de novo therapeutics development for NGLY1 deficiency, partnering with some medicinal chemists at the University of Utah, which is my institution at the time. And the first thing we did was we built a worm model of this disease. Because that's what I realized about biologists. They like to take other animals and just give them human diseases. It's a thing they do. And so we took planarian worms, and we chose them for a very specific reason. We wanted us to find something to address the really awful developmental aspects of this disease. And the thing that's awesome about planaria is that if you take a planarian worm and you cut it in half, what you get is 2 planaria. So they're very good for studying developmental disorders because they have this effect. Now, the interesting thing is, if you take a planarian worm and you cut it and you give it NGLY1 deficiency, in this case we did it through a knockdown with RNAi, and then you cut it in half, do you know what you get? What you get is a worm that's been cut in half. So it completely loses this ability to regenerate. So we're like, okay, we've got exactly the phenotype we're looking for. We want to try to reverse this phenotype chemically or genetically. So then what we started doing was knocking out other genes to see if knocking out other genes would actually reverse this loss of regenerative phenotype. And eventually, we actually found one. Actually, through some very educated guessing, looking at the literature, we were able to very quickly find another gene that, through its elimination, seemed to reverse this developmental phenotype. So if you knock out both NGLY1 and NGAs, these worms actually start to regenerate again. And this is actually something that it surprises computer scientists, but it doesn't at all surprise biologists, because biology is super weird. So it is literally the case that missing 2 genes is better than missing 1 gene. That's what we're saying here. So I don't know, we're sort of understanding why this is true now. But in some ways, we still don't really understand why this happens. But actually, in drug development, they do this all the time. They'll start randomly knocking out other genes and go, well, does this make a difference? If it does, they go, okay, well, now we'll just find an inhibitor for that. And they often never have a mechanistic explanation for why it seems to make a difference, and they don't care. They just want to find the inhibitor at that point. So we know at this point, what we need is an inhibitor for gene X, which is NGAs. To do this, to find this inhibitor, I went from biology and medicine back to computer science. And for this, we took a bacterial model of this other protein, NGAs. Now, because there wasn't a crystal structure available for the human version. So we take this, which is the bacterial version, and we use a process called homology modeling to compute what we think is the human version of the crystal structure of NGAs. And so we've got that. And then there's some pocket on this molecule, it's catalytic domain, where everything actually happens, which actually makes the reaction go. We have to find some small molecule that is inverse in shape and charge to that catalytic domain. That is the computational question we're trying to answer. So you can do a bunch of simulations called docking simulations, and you can find molecules that fit right in that domain and look like they might be inhibitors or at least interactors with something like NGAs. So we looked at 200,000 compounds in silico. 70 compounds came out as potential inhibitors for NGAs. 14 of them were already FDA approved. So we took those 14 to a lab. We used mass spectrometry to interrogate whether or not they really were NGAs inhibitors, and we found out that one of them actually was. And that one turns out to be Prevacid. So Prevacid is like an acid reflux medication. Now, Bertrand doesn't have acid reflux, but it doesn't matter because there's a weird side effect to Prevacid that we just discovered. It's an NGAs inhibitor. So if you take Prevacid, you're inhibiting NGAs. You didn't know that, and you didn't care. But we care a lot because this is how you actually disable that gene, which appears to reverse the phenotype for NGLY1 deficiency. So if you want to know more about this, wrote up and published it recently in a paper. And the one thing I'll say here is even though I spent 10 wonderful years in computer science, I think this is my favorite paper of all time. Because this is a paper, and it's not a computer science paper at all, but this is the paper that describes a viable treatment for my son. And then, I remember when it was getting diagnosed 5 years ago, the researchers were like, yeah, and no drug company will ever take an interest in this. Except that the science has been so thoroughly de-risked that Retrophin actually had the patient community out to meet with them and say, we think we can take Prevacid and make a super Prevacid, something 1,000 times stronger than Prevacid so that you don't have to take it at toxic doses to get this effect. And so what we're doing now is we're taking the core scaffold for Prevacid, actually all the proton pump inhibitors, of which Prevacid happens to be, and modifying it and making lots of small tweaks to see if any of these actually give you a better Prevacid. And we're already up to something like 60 times more potent than Prevacid, or the best proton pump inhibitor we found so far, while still inhibiting NGAs. So we're on the way towards a super Prevacid, because right now, it's an inhibitor for NGAs, but it's not strong enough to inhibit it fully at non-toxic quantities. So he's on it now. He's getting some inhibition of NGAs, and we're definitely seeing some benefit to him, but we know we can do much better. We know we can build a new drug based on this to do it all over. So the question is, will this work again? Can we actually repeat this process of finding treatments for in light of individual mutations? And the reason I know the answer to this is yes is that I got some really unusual opportunities for which I am truly super grateful. So the first one was the Harvard Med School said, hey, we see what you're doing with your son. We'd like to know if you could scale that up for other patients. Why don't you come join the faculty here? So I began a visiting professorship there almost 3 years ago. I'm still there today. And then the next year, this guy said, why don't you come to my house and let's chat? So I get this call. I go out there. It's Obama. He goes, hey, I heard what you did for your son. I'd like you to do that for the rest of the country. And I go, okay, anything you want, obviously. I mean, you don't say no to Obama. So that started my engagement with the precision medicine initiative. This was actually before it launched. So I wrote some of the founding white papers around how we're going to organize this thing, what kind of science we're going to do, and acted sort of I guess maybe an architect, if you want to call it that, of this initiative early on. And then I actually, I was going out to the White House quite regularly at that point, and then I actually just joined the White House as an official. And so shortly before that, I actually announced with President Obama the launch of this new initiative to sort of do a proof of concept for precision medicine called PEPMA, or the Patient Empowered Precision Medicine Alliance. And the whole idea behind this was we wanted to see, could we really do the whole NGLI1 story again really quickly for a number of other diseases? So we picked 5 diseases and said, can we find viable clinical candidates for treatments in just 12 months? The reason 12 months was important was that was all the time that Obama had left in office. And so that bounded it, it very naturally bounded the problem. The thing is, we actually succeeded. We actually did that. So I stitched together this series of institutions, and this acted as essentially the processor to execute that algorithm that I showed you earlier. This was our computational substrate for executing the algorithm for precision medicine. And so I was actually able to go back shortly before he left office. I think this was like 3 or 4 days before he left, and say, yeah, hey, we did it. We have proven that precision medicine actually works. We can do this again because we've done it now for several other diseases. What I will tell you is that I very carefully picked those 5 diseases. I didn't take 5 diseases at random. So not every disease is created equal in terms of its ability to find a treatment. However, I'm finding that large regions of the genome can be divided up according to essentially their screenability for drugs. And so depending on the gene you care about, you might actually have a pretty easy path to finding a way of interacting with that particular gene. So the other thing I realized very quickly is that, throughout the past 9 years really, is that this is all about a lot more than just computation, even about a lot more than just science as well. Because as hard as it is to get a diagnosis on something like precision medicine, although that's getting easier and easier, and as difficult it is to find a therapy, this new mountain appears once you climb those 2, which is actually getting it past the FDA. So regulatory approval is more challenging even still. So to address this is something else I encourage people to do. So I've actually I've spoken to Congress and the Senate when I was allowed to do these sorts of things, and I urge people to always contact their legislators and say, the science is way ahead of the regulation here. We need better policies for getting drugs approved in things like precision medicine and rare disease and in cancer, because the patients need these, and we can find them for them now. So what's next? Well, like I said, I'm actually still there at the White House, so I'm doing the best I can to argue for precision medicine and science in general. That's been interesting. And I can't comment anymore. So then I got sort of the offer of a lifetime. I didn't really see this coming, but UAB, the University of Alabama at Birmingham, said, we would like you to serve as director of this brand new precision medicine institute that we're setting up. And I said, okay. The amount of resources, so basically this offer is they said, come run this institute, hire a faculty, hire professors, create a vision for what clinical precision medicine is, and develop a research program that enables that. So that's what I said yes, and actually that's what I've been doing since July. So I'm now down there at UAB as director of this institute and seeing patients, which is crazy because my PhD is still in computer science. and mapping out a vision for how we deliver clinical precision medicine. So I've come up with 3 focus areas. Obviously, rare disease is close to my heart, so we definitely have that as an emphasis. But then precision oncology, because I think if we're going to go after diseases of the genome, cancer is the big one. That's the disease we really need to hit. And then for the rest of us, I'm offering pharmacogenomics. So this allows any of you to use your genetics to determine if you have a sensitivity to a particular medication, or if for any particular disease there's one medication that's much better than another. So this is the update. I think the last time I gave a talk like this was in Barcelona, and I hadn't actually started that job yet. But now that I've been there about 5 months, I can tell you some of the things that I've been doing. So for handling rare diseases, I've partnered with this great guy here, Bruce Korf, who already had an existing undiagnosed diseases program there. So if you have an undiagnosed disease or know somebody that does, you can send them to us. We will sequence their genome. We will try to find the mutation driving their disease. Once we find that cause, we can then use another company in Birmingham called Southern Research, which is essentially a disintermediated drug development company, and we can use robots to test thousands or even millions of compounds against their cells or models of their cells or models of their disease to see if anything works for them. So we have both a diagnosis program and a therapeutics program running side by side now at UAB. And to do this, I'm booting up what I'm calling the Human Screenome Project. And the way this works is we're coming up with a set of broad, very reusable assays, some of which are actually computational, that give us very high coverage of our ability to modulate the human genome. And so what we're really doing is we're dividing up the human genome into regions of behavior or types, and then saying, okay, well, this assay, which is a test for looking at what a particular cell or model system is doing, can interrogate the behavior of these genes. And sometimes 2 assays will both work on the same gene. Some assays only work on 1 gene. And then so what happens is a patient walks in the door. We use sequencing to find the gene that's misbehaving in them. We use this database then to find an assay that can look at what that gene is doing so we can study it in a dish. And then we can take that to a high-throughput screening facility and say, does any drug now make a difference for this particular gene so that we can find a drug to give to a particular patient? And the patients are actually showing up, and we're actually doing this kind of stuff. So just to give you an example, I've got a patient that showed up on I think the 2nd week I was there with a mutation in NTRK2. Now, we knew this was the cause because we found, I think, 4 other patients that had mutations in this gene. This patient, I should actually say, was actually found with a joint collaboration through another institute called Hudson Alpha that's also in Alabama. It's also using sequencing to give diagnoses to patients that are undiagnosed. And this is the particular mutation we found. So at the 434th codon, there was a substitution of a tyrosine for a cysteine. That's this little patch notation or diff notation that we use for encoding mutations. And it happens to occur in a transmembrane domain, so we think it was causing loss of function, but maybe it's doing gain of function. So the question is, how do you treat? What we have to do is instantiate the algorithm for precision medicine that I showed you earlier for this particular patient. And so the algorithm, when we specialize it now for this patient, looks like this. We sequence, which we did. Then we do analysis. Is it gain of function? Is it loss of function? If it's a gain of function, we have to find an inhibitor. It's a loss of function, we have to find an activator. And so what I do as director of this institute is I go across UAB or outside UAB if I have to, and I map people onto the phases of this algorithm and say, you're the one that's going to do this phase. And so I found somebody to do the analysis of this gene, and I already know if it's a gain of function. I found somebody who can find the inhibitor because it's actually a gene involved in cancer, so there's cancer companies developing inhibitors for it. Then I found somebody else who has already developed an activator for this gene because it needs to be activated in a completely different disease. So whichever direction this analysis falls out, I've got something that I can offer to this patient now. The only thing I'm waiting on is the IRB approval to take a cheek swab from this patient so we can do the analysis. That's the only bottleneck. It's not the science, it's the regulation. And IRB, as I'm learning, can take forever to set up. So once we find an inhibitor and an activator, we can do some medicinal chemistry to optimize it potentially if we need to, then move on to a clinical trial for an N of 1. We've got another patient that's got epilepsy that we did a limited sequencing on. We found 2 variants of uncertain significance. And so now we go, okay, well, which mutation is really driving this disease here? We've got to figure out which one it is before we can come up with any kind of treatment. And this is actually cool because we did some computational modeling. So the 2 genes that popped up here were KCN5H, which is a potassium channel, and RYR3, which is a ryanodine receptor. And we said, okay, which of these is it? So to figure this out, we did computational modeling of these 2 structures and zoomed in on RYR3 and found that it looks like you get some transient loss of function from the mutation occurring here just by looking at the effect on structure. So by looking at this effect on structure, we can say this is probably the cause of this patient's epilepsy. This is the gene we have to target. And so that's where we are now. So now we'll do potentially docking simulations, see if there's any way to interact with that particular mutation. So now, what about cancer? Well, I've got a deputy director, Eddie Yang, running our precision oncology program. And one of the reasons I loved Eddie is that he was already showing me his algorithms for precision oncology before I even arrived at UAB. So we've been developing these algorithms, making them more complex, so that we can go from patient data, or really your tumor data, because we sequence the tumor and not just you, and figure out medications that precisely counteract the effect of the mutations in your tumors. So that's what we're doing now. And the great thing about these, so we've got 2 clinical trials running, and the thing is we don't care what kind of cancer you have. We don't do just breast cancer or brain cancer. We'll take any cancer, because all cancer does is now just mutations in a genome. And we're finding some interesting things across all kinds of cancers, so that there are some mutations that show up again and again and drive the vast majority of cancers. And what's even more remarkable is that for 71% of the patients that have walked in the door in these trials, we have found a matching compound that, at least in theory, will interact with the gene that we need to for their particular cancer. Now, we'll know in about 6 months to a year what this means in terms of survival and outcomes, but we're now, through these trials, directly addressing the root cause of cancer. Whereas chemotherapy is more like carpet bombing. With chemotherapy, you're really trying to kill the cancer faster than you're killing the patient. That's the goal there. Here, we're trying to go to the root cause of the cancer and say, this is the mutation driving your cancer. This is a drug that interacts with that root cause. Let's see if it works. Now, for patients that go treatment resistant, we have a separate project. Right now, we're doing it for leukemia. And what we'll do is we'll take patient blood, and we will screen it against the full FDA-approved library to see if there's anything out there that will work with your particular leukemia. And in every case so far, we are finding something that works. So even patients that have lost every other option, there's something we can do for them. So what about common disease? What about most of us? Because most of us don't have disease. Most of us don't have cancer, not yet anyway. What about common diseases? Well, we're going to start tackling these one by one. And the first one I've picked is actually depression. So depression is a very widespread disease, has enormous societal costs and human costs. And the thing is, we can do a lot better than we currently do right now. So right now, if you get depression, you go on this trial and error process where you try all these different antidepressants, one after another, hoping something will finally work for you. This can take years as a therapeutic odyssey. What we can do instead is we can look at the genes that control metabolism of these antidepressants. We can say, well, you should never take this drug because this is really awful for you. Or you should probably be on this one instead because it's going to work great, or probably should. And for patients that adhere to the recommendations on this treatment protocol, the remission rates are up to 90%. So 90% experience broad relief from depression when we look at the genetic drivers of how you interact with antidepressants, if you can stick to the protocol, which is definitely a big if in depression. But this is significant because that's not the case when you don't have access to this information when you try to treat. So I would say in conclusion, as far as the war on error goes, that it's a difficult war, but it's one worth fighting in both people and in software, and it's one that we can win. And the core theme here, of course, is approximation. We can approximate solutions to the halting problem to predict the behavior of software, to find the flaws that drive most of what's plaguing us in software today. And with people, we can approximate solutions to what will help them, either in the forms of the set of pills that they should take or the procedures that they should do, or the set of experiments they need to run in order to find their treatment. So with that, I thank you. And if I have any time, I'd be happy to take your questions.
Some forms of cancer occur not because of the genetics, but because of the process of instantiating the genetics into cells. How do you tackle that?
Sorry, how do you tackle what?
When you take the genetics and you instantiate those into cells and that process fails.
Oh, I see. So the question is, you've analyzed a patient, you found a mutation, and now you need to build a cell culture of some kind to analyze that. So there's a number of techniques that you typically use to introduce a mutation into a cell culture. There's really new stuff like CRISPR, which you can use to make a cell model. And you're right, sometimes it fails. Sometimes you introduce a mutation and now the cell dies. Well, we don't often encounter this case with people because if it kills a cell, it probably killed the embryo. And so in most cases, when you introduce the genotype into some cell line, it usually doesn't die. Not right away, at least. And so usually you get something. Now, one problem you can have is that it might grow very slowly. So some mutations do have serious effects on cell growth. So there's a couple workarounds for this, and one of the workarounds is cancer. So you can take a cancer cell line, which grows very well, and you can take that, the mutation, and knock it into that. And now you have a very rapidly proliferating cell line for a disease. Now, is this the perfect model to study? Well, no, because you're also studying cancer at that point. But in many cases, it's a workable model of the disease. So you can take a cancer line, and you can give it a second genetic disease. And now you have, hopefully, a viable model at the cellular level. If that doesn't work, for some reason, you can try to build an animal model. You can build a fly, a worm, a fish, a mouse, something like that, and study that instead.
Hi, thanks. So yeah, I've far too many knowledge from my family with rare disease, but one of the problems that happens a lot when you do that type of stuff usually is a problem of studying the enzyme, because the folding is still something we don't understand that well. Do you encounter problem computation problem for the folding of the molecule and changing?
So the question is, all right, you found a mutation, and you know this mutation is going to have some effect on the folding of the protein, right? So do you and protein folding is a hard problem, at least if you do it sort of ab initio. If you just take a sequence and say, give me the protein, that's really hard. That's a very hard problem. And there's several ways to get around this. So like in the case of NGAs, we needed a structure for NGAs. So we started with a structure for bacterial NGAs. The sequences for that are pretty close to each other. And so we could just tweak it, again, through this process called homology modeling and get it to the actual structure. And you can do the same thing if you have a structure for a human protein and you've now introduced a mutation. You can insert just that mutation into the folded structure, and then you can modify that. You can also do X-ray crystallography. You can just forget computing it and say, well, nature's the best computer we've got. Let's just look at what it looks like. So you can do that. Sometimes crystals, but sometimes it's really hard to crystallize a protein, so you can't really do that. Well, now there's this amazing new technique called cryo-EM, where you can just look at the protein. So that's what we'll do now. So actually, I'm starting collaborations to use cryo-EM to do variant interpretation for real patients. Where instead of trying to compute it or do crystallography, we'll just look at it under a very powerful microscope and say, well, what do you do? What is your actual structure? And so I think the deeper thing here with this question is that we're computer scientists, and we like to do everything with computation. And that was my bias 5 years ago going into all this. I was like, I'm going to do everything with computation, everything. And I realized very quickly that in many cases, the fastest computer we have is nature. That is way faster in some cases to put a mutation in a worm and watch what it does to the worm than to try to come up with this molecular dynamics simulation that will somehow magically figure out all the way back up to the cell what's really going on. That's hard. So I've been re-educated. And while I love computation, and I always will, I'm willing to concede that in some cases you just want to go to a lab and do something. And that's maybe the fastest thing we can do. And certainly that is the case here.
Thank you for a wonderful talk. Wish you a lot of success in the wars and especially the first aim in your war, of course, your first patient. I was wondering how much computer science research do you need in your current research? Which progress in computer science would help you?
That's a great question. So how much computer science do I need for what I'm doing in precision medicine? Well, I think there's two answers to that question. And the first is that not a whole lot of PL, which is unfortunate given what I spent 10 years of my life doing. But I think systems biology, which is kind of like PL turned into biology, is actually quite useful. And we can do, and so like for Kappa is basically using programming language techniques to make reasonable predictions about what's happening in a particular disease within a cell. So that's useful, and that is reusing some PL, and I think it will have some benefit. I think deep learning has a lot of promise when it comes to genomic and precision medicine. For things like variant interpretation, I think if we want to know what a mutation is doing, it's probably something we can learn. I think toxicology, of a small molecule is something we can do deep learning on that will broadly accelerate the entire drug development process. So going forward, I'm looking to deep learning as a big hammer for us. But more than any particular kind of computer science that I need for what I'm doing, what I really need are computer scientists. I think something happens when you take a computer scientist and you turn them into a biologist. That doesn't happen when you take a biologist and try to make them a computer scientist. You don't get the same thing at the end of those two processes. And unfortunately, most of the time, that's the direction you go as biologist to crappy Python programmer. That's typically what happens. And I'm telling you all right now, like, there are labs that are desperate for your expertise. You don't have to know a whole lot of biology to do a heck of a lot of good. Because, I mean, most of the code being written in biology is just awful. And none of the people in this room would ever write it. So you could say, if you go to a lab and say, hey, could I just volunteer for a little while as a bioinformatician? They will be, they will be extremely grateful for your help. And then you'll be horrified for a little while, and then you'll do good things. So everybody in this room, I think, has the potential to really help out biology just by volunteering your time to some lab and trying to solve some of their computational problems. And in many cases, they're not that hard. They're like basic programming things that make a huge difference to their research. The reason that they can't do this systematically is they can't afford us. When you get a research grant from the government to hire a biologist, this literally happens where if they paid the market rate for a programmer, then the professor would be making far less than the programmer. And so many PIs were just like, I'm not going to do that. And in fact, NIH won't even allow it. So they desperately need our expertise. They just can't afford it.